Welcome in the next lecture. In this one, we will be discussing about theories of organization of shoot apical meristem. Now, in the previous classes, we have discussed about shoot apical meristem and root apical meristem and how they are organized at their growing tips. So, this organization which we have studied, it's the work of several scientists which through a course of time they have analyzed these tissues by various means. They have done cytological studies, they have done histological studies, they have analyzed lot of different plant types, and from there they have gathered information and they have postulated some theories which will help us to understand the organization and development of these apical meristem. Now, different theories have been proposed. Now, these names are here. It's not, many theories have been given for both root and shoot. However, some theories are applicable, more suited for root, whereas some for shoot. So, when we will move on, I will try to tell you that which is best suited for which particular organization. Now, like apical cell theory is there, histogen theory, tunica corpus theory, corpor cappe theory. So, all these theories we will discuss in the course of our lecture. So, first of all, we will try to understand the organization of shoot apical meristem by understanding the apical cell theory. Now, apical cell theory was given by it was actually proposed by Hofmeister and it was advanced by Nageli. It is applicable for both root and shoot apex. Now, according to this apical cell theory, they analyzed or they identified that there is a single apical cell which is present in the apical meristem. Okay, that means a single cell is acting as an apical meristem, and this single cell is giving rise to all different parts of a shoot as well as root. Now, if we closely identify this apical cell which is present in the apical meristem, we will see a single cell which is actually tetrahedral in shape. Okay, so if we analyze these layers of cell which is present in the shoot apical meristem, we will find that there is a single apical cell which is located on top or terminal position, which is known as apical cell or apical initial. Okay, now this single cell will give rise to all other cells, and finally these derivative cells will form different cell types. Now, this single apical cell is observed that it is tetrahedral in shape. That means it has actually 4 faces. Okay, and this single tetrahedral cell is found in basically in lower cryptogams like pteridophytes, in equisetum and many ferns. Like in lower cryptogams, you have some examples are like ferns, you have equisetum is there, then Lycopodium, Selaginella is there. In that also in cell is formed, and this will give rise to all the other cell. Now, when we talk about shoot apical meristem and shoot, we try to analyze the apical cell of shoot. See, it is also, we can say that it is inverted pyramidal in shape, that the structure which we have drawn. See, it has 4 faces and it is all the rest. See, 3 faces are actually embedded inside this plant body, and 1 face will be on the top side, and this 3 phase, actually the 3 phases which are there, they will give rise to, they will cut off cells and will form new cells in a very regular fashion. Okay, there if we try to analyze this apical cell, now, the derivatives of this apical cell, they are formed in a very highly ordered fashion from the other three faces. Like first of all, a single cell will be cut off from this side, okay, then it will be followed by the next phase and then followed by the third phase. So, again this will cleave off new cells followed by, so a highly ordered fashion this entire cell division will take place on the three faces. Somewhat flat cells will be formed, and in some ferns, like still in some pteridophytes, water ferns, it is observed that the apical cell is three-sided, but this is not always true for every case. So, in this three side, only two sides will cleave off, whereas one side will be on the top, and in that a very symmetrical orientation, like one is coming out from this side and the other is coming out from this side. So, in this case a very symmetrical arrangement you can see, but this is not very common in all type of fern. You will have a tetrahedral structure only. Now, in Selaginella, which is also a pteridophyte, actually it is observed by various studies that this apical cell is actually not a single cell, it is two cells. It is arranged in, and there is an anticlinal, means there is a longitudinal division giving rise to two cells, but this is also not very common. It is found in Selaginella. Now, this figure shows a structure of equisetum apical cell. Now, equisetum is a pteridophyte. Now, in this case also you can see here that this apical cell is a single cell, okay, tetrahedral in shape. However, it may not be very clear, but from the arrangement of its derivative you can easily make out that this one present at the top is apical cell, because they are forming a highly ordered fashion of arrangement. Now, but this is applicable in lower cryptogams, lower pteridophytes, in vascular cryptogams in pteridophytes, and in that also in lower pteridophyte groups. However, multicellular shoot apical meristem is found in most of the organism, in most of the higher plants. It is found in angiosperm, it is found in gymnosperm, and even in some pteridophyte also multicellular shoot apical meristem is identified. Now, later, because this apical meristem actually they were not able to clearly define or clearly explain the organization of the complex structures which are present in higher organism. So, this theory was said that it is only it could only explain the vascular cryptogam, whereas for higher multicellular organism or for higher plants, new theories were proposed. So, the most important theory which was proposed is the histogen theory, which was proposed by Hanstein. Now, this is also applicable for root as well as shoot apical meristem, but still it is more applicable for root, although both the things could be explained on the basis of histogen theory. Now, according to this theory, the multicellular apical meristem is organized into different zones, and each zone consists of its own initial cell, and these zones are known as histogen. Now, histo actually means tissue, okay. If we talk about histo word, it means tissue, okay, and this is taken from generation, okay. That means tissue generation. The cells or the layers which are capable of generating tissue is known as histogen. Now basically 3 histogens were identified, so that also we have slightly discussed, but and that classification which we have discussed in root apices was actually based on histogen theory. So these 3 layers were identified: dermatogen, peribellum and pleurome. So if you try to see the structure of this shoot apical meristem, basically 3 layers were there. So dermatogen, you have peribellum and pleurome. So in sense of root apices we have discussed all those things, but in shoot also you can find 3 layers were formed. Dermatogen was giving rise to epidermis, peribellum was giving rise to cortex and pleurome was giving rise to vascular tissues. Habertland proposed names protoderm, ground meristem and procambium for these 3 histogen. See, protoderm, ground meristem, that also we have studied when we were studying about classification of meristem on the basis of their function. So these different layers name, same thing is there: protoderm which is giving rise to epidermis, ground meristem to cortex and procambium is giving rise to vascular tissue. So these are 3 histogen and 3 layers which all have their own group of initial cells. Okay, they are not linked, means it is destined that dermatogen will give rise to epidermis and peribellum to cortex and pleurome to vascular tissue. These 3 layers are committal, means they are destined to form these 3 parts of the root or shoot. So definitely the drawback of this histogen theory is that since the histogen was committal, means they have a fixed destination, but when later different groups of plant types were studied, it was found that you cannot just fix the destiny of these 3 layers, and it's not that in every plant type you will have these 3 layers. Like for example in gymnosperm these 3 layers, and in many angiosperms also these 3 layers could not be distinguished clearly that this which one is which. So it was very difficult, and the role of these 3 layers also could not be assigned to each of the histogen. So this theory was later replaced by tunica corpus theory, and which was proposed by Schmidt. And in this theory actually it is not the different layers, okay, the layers are differentiated on the basis of plane of division, okay, not on the basis of their destiny that this layer will form this particular type of tissue, but on the plane of division, that if one layer is dividing into a particular plane, that means it is given a separate name and it is given a separate function. Now according to this theory, when we try to analyze the structure of shoot apical meristem, okay. So according to this theory, basically this shoot apical meristem is divided into 2 tissue zones. The outer one is known as tunica and the inner one is known as corpus. So the superficial layer is the tunica layer and the innermost layer is the corpus. They could be designated into L1, and L2 and L3. Okay, so basically this L1 and L2 is the tunica layer and L3 is the corpus layer. Now the most distinguishing feature of these layers are that the tunica will divide by anticlinal division. Okay, so anticlinal and periclinal we have already discussed. Like periclinal means parallel to the surface. Okay, so if a surface, suppose this is the surface, okay, and this is a cell, it is dividing parallel. Okay, it is dividing parallel to the surface, so it will give it will add cells on both the sides, so it will actually increase the volume of the inner mass of this cell. Okay, it will increase the volume, and in anticlinal division it will increase the number of cell on the same plane, so it will increase the surface area. So the layers on tunica L1 and L2, basically two layers of tunica is generally found in most of the plant types which have been studied. So this L1 and L2 layers, they both divide anticlinally, so they will increase the surface, they will form a sheet like outer covering. So naturally the destiny for this L1 layer, it will be that it will give rise to epidermis. Now L2 layer they will also divide anticlinally, and sometimes they will form subepidermal structures, and it may also contribute to the cortex and vascular tissue. And corpus, here we can see that they are dividing in all planes, so they will increase the volume of the cell and they will give rise to all different parts of the cell, leaving the surface layer. Now each layer will arise from separate initial, means every layer will have their own separate group of cells which will give rise to each layer, in contrast to the apical cell theory where only one cell was giving rise to everything. In multicellular conditions, like in histogen theory also we have discussed that these zones have their own separate initial. In tunica corpus theory also these own different layers have their different initials, and each initial will give rise to their own cell types. Now generally the tunica is two layer, but in many cases even 4 or 5 layers of tunica has been observed, but 2 layer is the most common one. So the merits of tunica corpus theory is that, see, in histogen theory, the zones were committal, means they have a fixed destination, and that's why they were discarded in many cases. However, in tunica corpus theory, these layers are not so distinguished. They are only separated on the basis of their plane of division, and many a times the different layers can have can function to give rise to different cell types. This is a very simple, it's also thing is that it is based on a very simple explanation, and it explains the in a simple way it explains the organization of shoot apical meristem without any relation to histogenesis. That means without any relation to, you can say, the cellular characteristics, like what are the tissue structures, or what are the other parameters, like suppose dense cytoplasm or mitotic, means their division cycle, their growth cycle. So all these parameters are not considered, only they deal with plane of division. So this is one of the advantage of tunica corpus theory, and in many of the plant type, this theory is actually used to explain the organization of shoot apical meristem, and that's why when we were discussing about shoot apical meristem, on the basis of this theory only, we have tried to understand the organization of those layers. We can discuss about mental and core concept. See, this also is based on tunica corpus organization only, but yeah, definitely one drawback was there, that it was considering plane of division. Okay, and many a times certain plant species like some cryptogams and some gymnosperm, actually they do not show a tunica corpus organization because they do not have actually a shoot apical meristem. The surface layer, they do not show a surface layer which is dividing by anticlinal division. Okay, so if it is not dividing, we cannot say it as tunica according to tunica corpus theory, and therefore we cannot differentiate them into different layers. So Popham and Chan gave this mental core concept. In this, they considered that all the cells which are present on the surface are known as mental, means a sheet of cell which is present on the surface is mental, and rest of the thing is forming the core. So here this plane of division is also not considered. Now moving on to the next cytohistological zonation concept, on which many of our plant type meristematic tissue has been characterized, and different zones have been identified on those in those shoot apical meristem, and it was given by Foster. Now this concept is not only based on plane of division but also on cytochemical differentiation, means how the cells are also different with respect to each zones, only not on the basis of their plane of division but also on the basis of their cellular types and how they are dividing, their mitotic index, their extent of cell division, their growth rate, means their division rate. So all these parameters are also taken into consideration under this cytohistological zonation concept. So in this concept, that also we have studied before while studying the organization of shoot apical meristem, because this is also one of the important concept on which the zones have been identified. So in this also, okay. So on this, according to this concept, in the shoot apical meristem, we can identify the central zone, and then we can have organizing center, and then you have peripheral zone, and this is the rib zone. Okay. Now this central zone is basically made up of cells which do not have a very high division rate. So here we can say that the difference is there, that according to the division rate also we can classify those cells. The central zones, they are not dividing as such. When the new cells are required on the other meristematic zone, this will become active and they will give rise to those cells, and they will give rise to the cells of peripheral zone or rib zone. The peripheral zones are the zones which are present on the sides, and they will give rise to the lateral organs, to the leaf, to the axillary bud and the other peripheral parts of the stem, whereas the rib zone will give rise to other parts of the plant organ like pith. It will give rise to cortex. Okay, and the organizing center is also present just below the central zone, and this organizing center is responsible to control the activity of meristematic tissue. It releases lot of chemicals, lot of hormones, which controls the activity of the central zone. Now A similar type of concept was given by Buvat, a waiting meristem concept. It is also on the basis of cytohistological concept. Actually, in this concept also, different zones are there. You have peripheral zone, you have a central zone, but the name they have given is slightly different. They have given a waiting meristem name which is equivalent to central zone. Waiting meristem means actually they are waiting to be meristematic in activity. So, they are non-dividing cell, but they will become meristematic when required, and then there will be a peripheral zone whose name they have given as initiating ring, and the medullary meristem which is equivalent to rib meristem. Okay. So, this is all about the organization of shoot apical meristem. In the next lecture, we will try to understand the theories which are related to root apical meristem, and that will help you to understand how the root apical meristem is organized and how from a single cell their development has been gone through in multicellular organism. Okay. Thank you for today.